In the next 5 minutes, this video will provide an overview of how you can use Skyline PIMS to manage data in the cell therapy industry using a representative CAR-T manufacturing process that is shown here. We have set up a process definition quickly and easily using point and click for establishing parameters, unit operations and phases without any IT skills necessary or programming. This process definition will then allow us to add limits, enter batch data and perform analytics in a very quick and easy way. This particular parameter has been set up just by giving a name, a description, showing where the data is coming from, either manually entered, calculated or derived from other parameters, or queried in the event that we do have electronic data sources such as electronic batch records, LIMS, or data historians. In this way, then we can assess criticality of parameters. We can add knowledge via notes, attachments, and references, and we can start entering data verification settings for later batch data entry. In this way, we've set up a structure for storing and managing your data across your supply chain, whether you have CMOs involved or whether you're internal manufacturing. And we have the ability to set up a limited number of users across that supply chain. So in the event that you wanted to provide access to your CMOs, they can be given roles as collaborative or as constrained as you wish. Moving on to batch data, we are looking here at a set of manufactured batches. We're going to look at one that is partway through the approval process. And we can see here that the process definition has been used to generate data entry fields, which can include warnings, it can include mini audit trails and embedded calculations, as well as tables for timestamp data. We can also look at metadata associated with this batch. In this case, we have the source of record, a PDF of the batch record, which can be inspected for deviations and quality investigations. In this way, we're able to approve data via a workflow and be able to manage data whether or not that data has been entered manually, imported from an Excel data sheet, especially for legacy data, or whether you've queried data from electronic data sources. We then have the ability to analyze this data. I'll make reference to a couple of different analytics capabilities here. This is looking at a set of batches with one critical parameter, or we could look at an individual batch and choose to monitor several parameters looking for cause and effect. We can trend any of the parameters that were originally set up within the process definition. Here I'm going to look at transduction efficiency, where some early process development batches have showed high degrees of variability. We've applied run rules, which can be automatically configured based on industry standards, and plot additional information such as release specifications, so that when we're tightening the variability for early phase clinical manufacturing, I've got the ability to inspect the variability of those batches, double click into an individual outlier, look at the history of the specifications, and in this case, pull out reference to a flow cytometer output. I can also save these charts to a variety of different file formats, and this way I can manage, share, and approve data in one centralized location with much higher data integrity, full audit trails, and Part 11 compliance. This can be done across the entire supply chain, using the Skyline's cloud-based technology and unique licensing model to work collaboratively with your CMOs and be able to accumulate a process history throughout the product lifecycle, whether it's for early stage clinical manufacturing or scaling up into pivotal and commercial manufacturing.